Hi, and welcome back to the Antibody Society's interview with the researchers behind the images that you have in your 2024 calendar. Today, I will be introducing our November image, and welcome to Virginia. So, welcome, Virginia. We are really happy to have you here as our November calendar participant. You submitted an absolutely beautiful image, and you've got a great response from everyone. So I wanted you just to introduce yourself and tell us a little bit about the research that you're doing.
Hi everyone, thanks Jessica. It's a pleasure for me to be here. I'm Virginia Metrangolo and I'm a first year postdoc. I recently got my PhD in cancer immunotherapy and I work in the Angelon group at the Finsen Laboratory, which belongs to the Biotech Research and Innovation Center in Copenhagen, Denmark. And my PhD was funded by the Marie Curie Fellowship under the Horizon 2020 program. And in my research group, we basically work on antibody drug conjugates for the treatment of several cancer types.
Brilliant. Congratulations on the PhD.
Thanks.
And hopefully we can look forward to some interesting information given that you're continuing on, so it must have been a very successful PhD.
Yeah, we are trying to, so the idea is that we would like potentially to spin the project out and let's see where we go from there.
No, that's brilliant. I know that's a lot of our people's aspirations, so it'll be great to hear from your experience.
So in this image, you can see that I was assessing basically the uptake of my candidate antibody that I later use as an ADC in pancreatic cancer cells. And I will explain you later on how the procedure works, but the antibody is labeled with a red fluorophore. So you can see these big dots in red. And then, of course, you can see the nuclei of the cells, and the surface of the cells is labeled in green. And you can also see that these cells tend to agglomerate and form these nice aggregates that are similar to pomeggranates. So it's a I wanted to start with a background on the mechanism of action and the drug concept of antibody drug conjugates. So just a second. Okay, so what are ADC? So I usually say that an antibody drug conjugate is a weaponized antibody. It consists of a monoclonal antibody that recognizes specifically a surface receptor which is overexpressed on the surface of tumor cells with limited or no expression on normal healthy cells. And when I say that the antibody is weaponized, it's because it carries a highly potent cytotoxic payload which is responsible for the cytotoxicity, and the linkage is provided by a plasma-stable linker, which ensures the stability of the complex into the bloodstream while enabling selective drug release of the toxin at the tumor site. And we normally say also that antibody drug conjugates work as Trojan horses, and the general mechanism behind ADC consists of a receptor-mediated endocytosis, meaning that after binding the antigen on the cell surface, the entire complex is internalized via receptor-mediated endocytosis. It ends up into the lysosome, where after processing, that can be either proteolytic processing or pH-induced release, the toxin is finally released into the cytoplasm, finally leading to cell death. And as you can see in this image, of course, the normal healthy cells which are devoid or express a minimal amount of the receptor should in theory be spared. And the idea behind this is that we try to supposedly increase the therapeutic window compared to conventional chemotherapy. And I also have to say that this field is actually actively growing. And as you can see from this image, that actually is a graphical abstract of a review recently published in January 2024. Here I try to represent what's the clinical landscape of ADC right now, but also with a view on the recent advances that are actually exciting developments in this field. So on the left, you can see that 14 ADC have been actually approved from 2011. And what is nice to see is that in addition to hematological cancer, several approvals have also been granted towards solid cancer types that are more difficult to treat, like breast cancer or colon cancer and urothelial cancer and so on and so forth. And on the right side, I just try to represent what are the advances and what will become the next generation ADC. And as you can see, there are several exciting developments spanning from the design and engineering of novel formats, linker technologies, payloads, and also the type of cancer cells we can target. And
that's a brilliant summary. So a lot of the ADCs are focusing on is the solid cancers. I know that you said that you were working on pancreatic cancer. Are any of these along those lines, or you'll be one of the first, hopefully, once it gets through the pipeline?
So I would say that our approach combines both conventional and also what is novel in the ADC field. So we have a conventional way of producing the antibody that in our case was the hybridoma technology, and now more and more we are going towards the recombinant production of antibodies for several reasons. So we just lay on a conventional way of producing the antibody. On the other hand, we explore a novel conjugation method that's a site-specific conjugation technology and also a novel emerging payload type. And also I would say that our ADC is able to target not only the cancer cells, but also the stromal cells. And this is actually quite nice and I would say new, because especially for immunosuppressive cancer types like pancreatic cancer and also other cancer types, for solid tumor types, the stroma is a relevant component that drives drug resistance. So it is becoming more and more clear that it is also a benefit to target both. So this both.
Yeah, I've heard it described as the stroma provides like a scaffold for the tumor itself, so you need to attack that as well. Would you agree with the analogy?
Yeah, of course it depends on the cancer types, because in some cancer types it has also been demonstrated the opposite, meaning that if you remove the stroma, then the tumor will spread more. So it depends. And of course, this requires several studies for better deciphering what is the immune landscape and the tumor microenvironment dynamics. But for pancreatic cancer, it's for sure clear that it is a highly cold immunogenic tumor. So targeting the stromal and the fibrotic component, I think that would provide a therapeutic advantage for successfully targeting this tumor type.
Brilliant.
And I wanted to also underline that there are key factors to consider for designing a safe and effective ADC. In addition to the three structural components that you can see here, another important factor is, of course, the target type. And these two components, so the target and the conjugate, need to be harmonically balanced also in according to the specific tumor biology, as I just mentioned, because there are cold, hot tumors, so we have to consider all these characteristics. And of course, these challenge us in the development phases of an ADC, because there is no one-fits-all solution. And importantly, when I say the target, we all know that the expression profile of a target is clearly important for defining an ADC target, meaning that we should have mostly a tumor selective expression with limited expression on normal tissue. But it is also becoming increasingly clear that another key crucial factor to consider for selecting either an antibody or an ADC target is the internalization. And we all see from the past, from the initial developments in the ADC field, that several candidates were clearly failing due to the use of poorly selecting antibodies, meaning that in the beginning, we tried to reuse some antibodies that were used as functional antagonists for ADC, but that doesn't work. So we need clearly, I think, to select, in my opinion, antibodies also based on their internalization capacity. And why do we think that ADC may have a potential in solid tumors? Well, of course, an ADC is still an antibody, meaning that it retains the normal functions of an antibody, for example, the antagonistic function of a surface receptor, but also the immune-engaging functions via the Fc part of the antibody. And this is for sure. But in addition to that, antibody drug conjugates are also able to deliver other mechanisms of toxicity. So not only this targeted tumor eradication via this payload release, but also another important mechanism is the so-called bystander killing effect. What do I mean by that? Well, antibody drug conjugates are very versatile. We can tweak the properties of the conjugates and, for example, select payloads that are able to diffuse out of the membrane of the targeted cell into the tumor milieu and target also the surrounding cancer cells in a target-independent fashion. And this is extremely important in tumor types like pancreatic cancer, but also other tumor types with heterogeneous target expression, because in this case, we can amplify the anti-tumor response. So it's very important also to rely on this orthogonal mechanism of cytotoxicity.
Does the environment around your target cell affect how far this passive toxic diffusion can go? So can you make it spread further, say in a tumor microenvironment, and it would be less spread in a normal environment to help with this just by the type of cells that are around?
Okay, this is a very good question. I think that it depends on the tumor type. Of course, in the case of a very fibrotic tumor like pancreatic cancer, having a bystander killing effect is an advantage, but it also depends on the conjugate and the antibody properties. For example, having a very high affinity antibody in this case would not help, because most of the antibody would stick into the edges of the tumor. Instead, we need something that goes inside. So maybe just tweaking the properties of the antibody and selecting an antibody with a medium affinity for the target, that would help to just let the antibody penetrate and go into the tumor. I also saw some beautiful studies where they try to couple to combine the ADC with an antibody first. So the antibody will bind to the edge of the tumor, allowing the ADC to penetrate further inside. So there are several studies into this. And of course, it also depends on, for example, in our case, the target is present both on the stroma and the cancer cells, and that also helps because also the stromal cells can drive this bystander effect. So it's a combination of both. I would say it depends. It's a case-by-case scenario. Imaging is quite important because we need to assess the internalization and the uptake of our candidate ADC antibodies in our cells. And in the image I show you, I use a confocal microscope, and now you will also see other images taken with the same microscope. So the way we do the experiment is there are several ways to do that. In this case, in particular, I incubated the cells for 4 hours with an Alexa 647 antibodies recognizing my target of interest. And then after that, I basically prepare the cells, I stain the cells for surface recognition, so for the surface and also for the nuclei. And then I just basically go to the confocal microscope, and these cells were fixed. But we can also perform live cell imaging staining. So we have several options. And in this case, it's a qualitative assessment because I just want to see if something goes inside, and then I perform a quantitative assessment by both flow cytometry and a live cell imaging system called IncuCyte. So in this experiment, it is important to include controls, negative controls, to basically confirm the specificity of the uptake. And in this particular experiment, I had the chance to work with PDAC cells which were knocked out for the target of interest. In this case, we have a control, so no antibody was present. But then after incubation for 4 hours with the antibodies, you can clearly see that in the cells expressing the target, we have the presence of these red punctuate structures, these red spots that indicate internalization, which are not present in the knockout cells. And of course, these indicate target-specific uptake. Also, another important thing to confirm in our case was the lysosomal trafficking of our antibody. And why is that important? It is important because most of the antibodies, as I showed you, most of the ADC, as I showed you, are processed into the lysosome. In our case, we use a lysosomal cleavable linker, so it was even more important. And to confirm the lysosomal trafficking, in our case, we use a lysosomal protease inhibitor, which basically prevents the clearance of the antibody into the lysosome by catepsins or other enzymes present in this organelle, leading to accumulation of the antibody at this side. And of course, after this, if it works, you will be able to see a more pronounced red signal, which was basically what we saw. So you can see that after incubation, co-incubation with E60 64D, we have the presence of these more pronounced red spots, which basically indicate that our antibodies successfully go to the lysosome. And this was an important confirmation of our of this lysosomal trafficking. I also confirmed this using a pH-sensitive probe linked to the antibody. So in this case, you will have release of fluorescence once at acidic pH. That's basically in the endosome lysosomal compartment. So these internalization stays are quite cool to perform, and also the results are beautiful to see.
Yeah, that's definitely beautiful. So this is without your payload attached. Do you see any difference in your images once you've actually attached the ADC?
So I have to say that I perform all the internalization assay in a non-ADC format with just the candidate antibody. It's also possible to do that with ADC. I've not done that. But I basically confirmed that the ADC was going into the lysosome via other means. So, for example, when I set an efficacy testing in vitro, I also use the same inhibitor 64D, and in this presence, the ADC should not be effective. So it is an indirect confirmation.
Cool. Yeah. No, so how about, you mentioned before with your mice that one of the differences is that we don't have the scaffolding in these images that we're looking at, correct? Have you looked at those from the grafts and things that you've looked at your in vivo work? And does it differ in the distribution of your antibody when you're looking at those sort of tumors?
Unfortunately, I've not performed any kind of distribution studies due to also to time constraints, but it would be very nice. It's something we are planning to do to basically couple the antibody with either most probably a radioactive label would be easier, because then we could perform PET. In my case, the only thing I did for the imaging in the in vivo setting, I just use luciferase transfected cells, pancreatic cancer cells, just to visualize the tumor itself, because it's very deep in the abdomen, but I've not performed any kind of distribution studies. But it would be very nice to do that, because that gives you some kind also information or how the antibody is distributing, and if healthy organs are also if the antibody is also going into healthy organs. That's also important for evaluation of toxicity. What we do is that we do a post-term analysis. So once we sacrifice the animals after the end of the study, we study the expression of our receptor, and also we are in different healthy organs, bone marrow, heart, kidney, liver, and so on, and also in the blood. And we also study the histological morphology of these organs after treatment to see how it goes. Yeah,
that's very thorough. No wonder you haven't had time to do everything. You're like, looking at everything from your previous experiments. I think that's an amazing story. And you mentioned to me earlier that it will hopefully be published soonish.
Yeah, we hope so.
We all wish you luck, and really look forward to reading the full story and learning some more, hopefully.
Thank you, Jessica.
No, thank you very much for joining us.